The topic discussed here is primary cell culture. Isolation of mouse embryo for primary cell culture. The procedure starts by induction of estrus. The detection of vaginal plug is noted as day 0 and the development of embryo is timed from that date. The dissection date is planned based upon the development of the embryo. The optimal age for preparing primary cultures from disaggregate embryo is 13 days, while for organ culture it is 13 to 14 days. A full-term embryo can be obtained by 19 to 21 days. The uterus is dissected out under aseptic conditions and transferred into DBSS solution. The embryos are then dissected out of the uteri. Isolation of chick embryos for primary cell culture. The fertilized eggs are incubated at 38 to 39 degrees centigrade. For obtaining dispersed cells from the whole embryo, dissection is planned on the 8th day, while for isolating organ rudiments on 10th or 13th day after incubation. Dissection should be done in aseptic conditions. The selected eggs are swabbed with 70% alcohol. A crack is made on the top of the shell and the shell is peeled off to the edge of the air sac by means of sterile forceps. Then the white shell membrane is peeled off gently to expose the cam membrane and the blood vessels. The embryo is then gently pulled out by the head by means of sterile forceps and transferred into DBSS solution in Petri dish. Here shows removing of chick embryos from the eggs. After dissection of the embryos, the fat and necrotic tissues are removed completely and the tissue is chopped finely, causing minimum damage. The enzymes that are used for disaggregation of cells are then removed by gentle centrifugation. The cells are then plated for primary cell culture. The concentration of the cells for primary cell culture should always be higher than that for the subcultures because a lot of cells lose their viability. The embryonic tissues disaggregate more readily and yield more viable cells. The medias that can be used for the culture are Eagle's media, Ham's F12 media, depending upon the requirement of the nutrient compositions. If serum is required, the fetal bovine serum is preferred over calf or horse serum. Isolation of specific cell types probably require selective media. The dissection or disaggregation of embryos for primary cell culture can be done by fine dissection, mechanically or by means of enzymes. The embryos can be finely chopped and then transferred to culture media or by mechanical disaggregation by sieving and pipetting. Enzymes like trypsin and collagenase are also used for disaggregation of cells before being transferred to culture media. Disaggregation of tissues by fine dissection. In this method, the tissue after being washed with DBSS are chopped into small pieces, approximately 1 mm cubes, and then transferred to centrifuge tubes. The tissues are washed 2 to 3 times by resuspension and settling. The tissue is then transferred to cultured flask and spread evenly over the growth surface with limited amount of growth media. The culture flasks are incubated overnight at 37 degrees centigrade. If the pieces show adhesion, the nutrient media is added gradually over 3 to 5 days to the usual volume. The flasks are monitored every day for its growth and checked for contamination and nutrient media. If outgrowth of cells are absorbed, the cells may be subcultured. Advantage of fine dissection method is when there is only a small amount of tissue, such as for skin biopsies, where mechanical and enzymatic disaggregation may lead to losing of some cells. It is practiced mostly in embryonic cells which can migrate out successfully. The major limitations of this particular method is poor adhesiveness of some tissues and the selection of cells in the outgrowth. The mechanical method of disaggregation of tissues for primary cell culture. In this method, there are four approaches. First, the scrapping or spillage method, where the cutting action or abrasion of cutting surface releases the cells. Next, the tissue samples are aseptically passed through a series of sieves which gradually reduce in mesh size. This leads to disaggregation of the tissue. Another way is by forcing the tissue fragments through a syringe or by repeated pipetting. The main disadvantage of this method is it may cause cell damage. The enzymatic method of disaggregation. The cell adhesion is due to the presence of a variety of homotypic interacting lipopeptides which are calcium dependent. Hence, they are sensitive to the chelating agents like EDTA. The intracellular matrix and the basement membranes contain glycoproteins and proteoglycans which are sensitive to many enzymes like trypsin, collagenase, elastase, dispase, to name some. These can be used for enzymatic disaggregation. The two important enzymes that are used in tissue disaggregation are collagenase and trypsin. Disaggregation with trypsin is known as trypsinization, and it can be used in two ways, that is warm trypsinization method and cold trypsinization method. Tissue disaggregation by warm trypsinization method. In this method, the dissected tissues are cleaned of all the necrotic and fat tissues and chopped into small pieces of 3 mm cubes. These are then transferred into pre-weighed vials, washed 2 to 3 times by resuspension and settling by sterile DBSS. The tissues are then transferred into trypsinization flask containing DPBSA and 2.5% trypsin. Trypsin can be used alone or in combination with other enzymes as per the requirement. The flasks are then incubated at 30 degrees centigrade with continuous stirring. Every 30 minutes, the stirring is stopped and the tissue is allowed to settle. The supernatant with the cells are collected in pre-weighed vials and chilled. To the remaining tissue in the flask, fresh amount of warm trypsin is added and stirred at 37 degrees centigrade. The process is repeated until complete disaggregation of the tissue occurs. Finally, pool the chilled supernatants and the cell count is taken and viability of the cells are checked. The cells are then diluted to the required concentration in the growth media and plated in cultured flask. Trypsinization with cold pre-exposure. The initial steps taken for washing of the tissues are similar to that discussed earlier. After washing is complete, the tissues are transferred into the empty trypsinization flask and then it is incubated with 0.25% of trypsin solution at 4 degrees centigrade overnight. After incubation with trypsin at 4 degrees centigrade overnight, the trypsin is carefully discarded, leaving the settled tissue and residual trypsin. This is then incubated at 37 degrees centigrade for 20 to 30 minutes. Warm trypsin is added and by pipetting, the tissues are broken down or dispersed into the solution. The cell suspension may then be filtered through sterile muslin or Steiner mesh. The cell count is taken and the viability is checked. The cells are then diluted to the required concentration by using the growth media and seeded into culture flasks. After tissue disaggregation, the cell count in the suspension is taken. The cell viability of the cells are checked by using certain dyes. The most commonly used are fluorescent diacetate and trypan blue. In case of fluorescent diacetate, the viable cells would release fluorescence, while in case of trypan blue, the non-viable cells have a blue cytoplasm. There are various other probes or dyes which can be used to check the cell viability of the cells after tissue disaggregation. The table here mentions some of them with their principles. The cell viability is checked and the percentage of cells viable in the culture is calculated. The cell viability can be checked by using a combination of dyes. Example, in this case, 3 dyes are used. Here, the calcine AM dyes stain the live cells, which look green in color, while propydium iodide stains the dead cells, which look red in color. The third dye, the Holist 33342, stains all cells, which look blue in color. Lymphocyte cultures as primary cell cultures. These are suspension cell cultures. For this, first the peripheral blood is drawn into sodium heparin coated tubes to prevent coagulation. They are mixed well and then transferred into centrifuge tubes and centrifuged at 2000 rpm for 10 minutes. This leads to the formation of 3 layers. The upper plasma layer is pipetted out. The buffy coat layer, which contains the WBCs, are retained and expelled into the nutrient solution CRPMI. Dilutions of this WBC suspension is done by using the nutrient media CRPMI. The cells are then made active for cell division by addition of PHA. Tighten the cover and then slightly allow CO2 to enter the flask. The cells are then incubated under aseptic conditions. The animal cell cultures are maintained in aseptic conditions in carbon dioxide cell incubators. The growth media and the pH is maintained. Certain medias that are used are DPBS, HBSS, PBS, and EBSS. The cell cultures incubated in aseptic controlled conditions proliferate and reach confluence. In this state, the cells need to be subcultured. Factors that indicate the need of subculturing are drop in pH, cell concentration, cell types, and morphological deterioration. There would always be a drop in pH when the cells rapidly utilize the nutrients and release metabolites into the media. This results in lowering of pH. Different cells have optimum pH for their growth. Example, the fibrinoblast cells grow at optimum pH 7.4 to 7.7. For epidermal cells, it is 5.5. Certain indicators like phenol red are commonly used to check the pH. The table on the right shows the different colors corresponding to different pH in the media. Generally, at pH 7 to 6.5, the cell stops growing. Higher than 6.5, the cells start losing its viability. The other factors that are required for subculturing are cell concentration, cell type, and morphological deterioration. High cell concentration exhausts the nutrient media. Hence, subculturing need to be done. The transformed cells require more frequent subculturing than the normal cell type. With time in culture, the morphological deterioration of cells also occur, like granularity around the nucleus, cytoplasmic vacuolation as shown in the figure below, and rounding up of cells with detachment from the substrate. For subculturing of monolayers, the cultures are first treated with pre-warmed trypsin EDTA to disrupt or disaggregate the cells. The steps later taken for subculturing have already been discussed in the earlier session. Thank you.